And we always have some people come a little bit late, and you can't exactly know when they're going to come or precisely how late they're going to be. So, you should feel free whenever you feel comfortable, or start whenever you feel comfortable doing so.
Okay. Well, how about I start now because the beginnings, I'm just going to go through a little bit of background into kind of my system and my big questions, and then we'll get into the part of this that I know nothing about that I'm really hoping that I can get input from people. And obviously, like I think one of the nice things about this seminar series being kind of lab meeting is interrupt me along the way. If there's any questions that come up or if you think I've said something that's gobbledygook. Okay, so I'll get started. This is kind of the context about why I'm doing the work which I am doing. So I'm at an interesting place called the Jean Mayer Human Nutrition Research Center on Aging. And it's a USDA owned building and facility. And we're kind of on a mission to understand relationships between nutrition, diet, and healthy aging. And I'm the eye person at the center. So I'm really particularly interested in age-related eye diseases and the potential role for diet and nutrition in modulating those. And one of the areas that I've been interested in for a long time, and this goes back to work that my previous mentor, Alan Taylor, had started in this group, was looking at dietary patterns and their relationship with age-related macular degeneration. And what we found through a lot of epidemiology is that dietary quality has a big impact on your risk for macular degeneration. So individuals that consume a high glycemic index diet or a Western diet have increased risk for macular degeneration. And now it's pretty well established that the opposite healthy dietary patterns, like a Mediterranean diet, can be protective for AMD. And we wanted to really try to, rather than tease out the epidemiology, which runs into its limitations and what we can do to really test that, we wanted to use a preclinical system to try to understand more mechanistically what was going on with this relationship. And so I'll go through this quickly because I've presented this a lot for several of you, and I don't want to bore you too much. But what we wanted to really do was set up a mouse system to look at diet, and specifically we wanted to address dietary carbohydrate quality. And the way we did this is that we used initially wild type mice that we aged to 12 months, and then we fed them two different kinds of diets. We fed them a low glycemic index diet and a high glycemic index diet. I'll use either LG or LGI or HG for those. And the way that we made these diets is they're identical in terms of all of the macro and micronutrients. The difference being the cornstarch that's in them. So the high glycemic index, all of the cornstarch consists of 100% amylopectin, which is rapidly digested postprandially into glucose and causes a high postprandial glycemic excursion. Long-term consumption will lead to hyperglycemia. Whereas in the low glycemic index diet, we played around with the starch composition such that most of the starch is consisting of amylose, which is it's broken down to glucose much slower, kind of like one glucose molecule at a time. It's a linear chain of glucose molecules, leaving some of that to actually get fermented in the colon as well, which will be important in a couple of slides. And what we showed is that if you feed the mice a high glycemic index diet and you age them, we need both of those things together. They develop an AMD-like retinopathy. And I'll show you a little bit about that without going into too much detail. But here you could see retinal degeneration, disorganization of photoreceptors, RPE, disorganization, and atrophy. Whereas the mice that we feed the low glycemic index to, or ones where we switch the diet from a high to a low glycemic index diet midway through the study, seem to maintain their retinal health throughout these studies. And we've actually tried to do a little bit of longitudinal analysis, not very thoroughly. But you could see that what happens is that with aging, the high glycemic index undergoes continual photoreceptor degeneration, whereas the low glycemic index takes much longer and then shows kind of a slower age-related photoreceptor degeneration. And we were interested in kind of exploring what's happening in this high glycemic index diet that's contributing to these degenerative phenotypes. I mean, we saw that there were a lot of lipid-like phenotypes in the RPE. And so that's going to be the theme of this presentation is I'm going to be trying to talk about lipid biology, which is not my specific expertise. So please humor me about this. But there's a couple of things we saw in terms of lipid accumulation in the RPE. One of them is that we saw oxidized lipids and their protein adducts accumulate in the RPE. So here's carboxyethylperol, which is derived from DHA, 22 to 6, and 3 polyunsaturated fatty acid. And here's 4-HNE, which is derived from arachidonic acid. And you can see that both of these are accumulating in the RPE of the high glycemic index diet, but not so much in the low glycemic index diet. And ultrastructurally, we see signs that the RPE does not look particularly healthy. These are kind of like mouse AMD-ish like phenotypes. So the RPE undergoes vacuolation, which is commonly seen in multiple mouse models of AMD. We see these large basolaminar deposits form. I should say that even though the examples I'm showing from the low glycemic index fed mice do not show these basolaminar deposits, they certainly get plenty of basolaminar deposits, which we see accumulating more in relationship to age than anything else. What we do see that's abnormal in the high glycemic index is are these lipid particles that accumulate throughout the basolaminar deposits. And other labs have shown that these are full of lipids, even though we didn't do the OTAP staining that would demonstrate that. We also showed, and you'll see why I get into this in a little bit, changes in the gut microbiome in mice fed these different diets. You could see midway through the study, the gut microbiomes here's from the high glycemic index and the low glycemic index diets are quite different. When we change the diets, we can actually see that the gut microbiome undergoes a transitionary state and then resembles the diet that it finished up on. So the mice that went from a high to a low glycemic index diet end up with like what looks like a low glycemic index microbiome. And we were able to identify microbiota that both associated with diet and that associated with development of this AMD-like disease. Some of these microbiota increase with a high glycemic index diet, and some of them increase in the low glycemic index diet. And I'll talk a little bit about microbiota-dependent pathways in a second. But that's the background to what the story that I want to tell you about, which is what's happening in our NRF2 knockout mice. So I think most of you are familiar with NRF2. It's an antioxidant. It's an antioxidant master regulatory gene. So under oxidative stress conditions, the NRF2 becomes stabilized through degradation of its E3 ligase KEAP1. And upon stabilization, it translocates into the nucleus, where it activates genes that are involved in various steps of detoxification. And we've been interested in exploring NRF2 for a couple of reasons. One of which was we were wondering if we can accelerate our model of AMD by putting in kind of this genetic form of oxidative stress. And also other groups had shown that there was retinopathy in NRF2 knockout mice. Although I think there's like a little bit of disagreement about the extent of retinopathy and when it begins. So what we did is we fed the mice the same high and low glycemic index diets. We took the mice to about 18 months of age. Initially, we wanted to age these mice to get to 24 months. But NRF2 knockout mice develop a number of other problems, including liver hepatocellular carcinoma. And so we knew that these mice were not going to make it to 24 months. Um, so we stopped the experiment at about 18 months and just looked at what was happening in NRF2 knockout mice compared to wild type mice that had been aged on high and low glycemic index diets to 18 months. And what you could see here is that at 18 months, the NRF2 knockout mice actually undergoes like a pretty extensive degenerative phenotype. So we see a lot of extensive photoreceptor degeneration, which we quantify here as more extensive than what we see in a high glycemic index fed mouse. Here's an 18-month wild type. It's worse than the low glycemic index, not hugely as dramatically as what I showed you at 24 months. So we do see this kind of accelerated disease development. We also, here's, of course, my worst possible example of RPE atrophy in these mice. I would say we didn't typically see this level of RPE atrophy, but we did see a lot of other defects, both in the RPE, in the choroid, and we saw extensive loss of the choriocapillaris. So the NRF2 high glycemic index are definitely showing an accelerated disease progression. And then the interesting finding from this study, we published the eye phenotypes here already, is that if we feed the mice a low glycemic index diet, we actually largely rescue the retina. And that's this yellow group over here. Again, not quite as well as we would see in a wild type mouse. But keep in mind, these are still mice that are genetically deficient in NRF2. And these are whole body knockouts that we've been working with. So this was a really, to us, like a surprising finding that just by modulating diet, we could really affect the eye health outcome in these NRF2 knockout mice. I mean, it got us thinking that the impact of the diet is more systemic and widespread than we had come to appreciate, and it has important neuroprotective roles, but we did not know necessarily what they were. And I'll just tell you that the way that this story ended in this publication is that we did find that a variety of NRF2 target genes that had thought to be genetically dependent on NRF2 are actually still expressed in the NRF2 knockout mice. And some of them were upregulated in the low glycemic index mice. So the idea being that there's actually some genetically redundant pathways to some of these antioxidants. And the diet could be doing that. But we had the strong sense that there was more going on than just some upregulation of antioxidant repair genes. And so that's where kind of the published story mostly ends, and I want to take you on some new experiments.
Hey, Sheldon, it's
Yes.
Jason. Can I
Hi,
ask
Jake.
a couple questions just
Yeah.
about the model overall? Sorry, I was driving before. So you went over the nature of the diet of being this high spike in glucose. I mean, by definition, a high glycemic index. Do these mice get fatter? Do they develop diabetes? What's their fasting glucose levels? Is this, like, give us a profile of how the blood sugars move throughout the day.
Yeah, no, that's really important. So yes, the mice do become obese. No, they do not become diabetic, but they do develop kind of a chronic hyperglycemia, insulin resistant kind of phenotype.
Well, I guess how is that not diabetic? Sorry.
It,
Yeah,
I mean,
you're saying by glucose tolerance testing, they're not diabetic? Is that what you're saying?
yeah, I mean, the glucose, I mean, they have elevated fasting glucose. It's not dramatically increased. It does eventually go back to baseline.
You mean within a 24-hour period, there's
Yeah,
definitely periods where it's at baseline, but it's a more prolonged glucose.
right, exactly.
Okay. And the and out of
We do,
curiosity, I mean, I know
yeah.
this is the model you guys have been working on, but do you see or has anyone done, for example, streptozocin or other, OB mice crossed with NRF2, and is is it the same phenotype, or is there something different about being frankly diabetic versus these temporary spikes in glucose?
No, I think that's important. People have made NRF2 mice diabetic. Um, I don't think they live very long when you do that.
Okay.
And that was that's one of the advantages, I think, to this kind of dietary induced hyperglycemia model rather than like a streptozotocin model, is that we can generally still age these mice out. The NRF2 knockout mice that are made to be diabetic, I don't know exactly how long they live, but I know that people have shown that they do develop in the eyes, diabetic retinopathy-like phenotypes. Um,
Thanks.
yeah, I don't remember how far out those mice have been aged to and studied.
Since we're asking questions, I wanted to follow up on that and ask whether, um, sort of blood sugar parameters, um, or I don't know, if you were to do a glucose tolerance test, how long glucose would be elevated. Is any of that changed by the systemic nerve 2 knockout?
No, no, we tested that, and that was actually a little surprising to us because other people have published kind of that NRF2 knockout mice have a different response to um, like some studies have shown that they're innately less glucose intolerant, which again, I don't know why they would be. We actually, no, we didn't find any significant difference between the wild type and the NRF2 in terms of their glycemic responses. And part of that could just be that this is such a long-term adaptation to these diets. Whereas like maybe in a shorter term condition, there would be a more pronounced difference in them.
For sure. And I'm really fascinated by this compensatory, whatever is going on is taking over nerve two's role. That's pretty cool in and of itself. I know that's not what you're covering now. It's just neat direction.
Yeah, no, I think it's surprising to think about like environmental non-autonomous impacts on this like whole body knockout of NRF2.
Sheldon, I'm
Okay,
sorry. I just
yeah,
another set of questions. Um, the basal laminar deposits that you see are mainly protein rich, or is there lipid in the basal laminar deposits? And
it
the vacuolization is all intracellular and presumably lipid rich. Is that correct?
yes. Um, Definitely the basal laminar deposits are both protein rich. I mean, we see like the kind of banded collagen patterns, but and lipid rich as well. Um, we can see that histologically. The vacuoles, I'm less sure about their being lipid rich. And we
Okay.
haven't been able to, I mean, we have a separate lipid droplet phenotype that I think is distinct from the vacuoles, which
Okay.
I don't know if those are lipid containing or not. Okay. So here's a segue to another topic. Um, And this was this interesting idea. So I kind of told you about changes in the gut microbiome. And there's the literature on gut microbiome affecting metabolism is vast beyond anything that I could cover in any finite period of time. Um, but there's also an interesting body of literature looking at what happens to changes in the gut microbiome to metabolism in the liver, and then how that relates to the brain and the eye. And what I want to do is I want to kind of highlight a paper from Marie-Agnès Briget, who um, has done some of this work. She's one of ours. So I'd like to think about some of her work and how that's made us think about what could be going on in our models. So this is a paper that she published where what she did is she took germ-free mice and then reconstituted their microbiomes with microbiota from either young or old mice. And people have kind of done these experiments in a variety of parameters and found that if you reconstitute the microbiota with the gut microbiome, even in a young germ-free mouse with old microbiota, they show certain accelerated aging phenotypes. One of the things that she showed is actually that you get this change in N3 PUFA productions when you give them old microbiota compared to young microbiota. This is measured in the liver, but she's also looked in the brain, and you could also see similarly decreases in DHA in those mice that are constituted with old microbiota compared to young microbiota. And when she looked at enzymes in the liver, you can actually see that the gut microbiome is affecting the levels of some of these key enzymes that are involved in production of omega-3 PUFAs. And this reminded us of something that we found in mice that had been fed a high glycemic index diet. This is actually work that we did in collaboration with Anand Swarup's lab, and everything I'm going to show you afterwards is kind of this continuing ongoing collaboration with Anand Swarup's lab. In this particular study, we compared short-term to long-term feeding of a high glycemic index diet. But we found something kind of that reminded us of what she characterized with her old microbiota, which is again like these increases in SCD1, ELO-L5, and that changed a variety of lipid composition in the liver. The most profound change, actually, I'm not showing in this slide, but I'll show you what happens later on, is that, as you might guess, with increased SCD1, we see a large increase in monounsaturated fatty acids, specifically 16:1 and 18:1 lipids, at the expense of PUFAs. But we also saw something similar to what Marianne had described, which was this drop in N3 PUFAs, and especially DHA, with a high glycemic index diet. We don't know if that also affects lipid composition in the eye or in the brain, as Marianne measured, but we have good reason to believe it should be, because we're not adding any exogenous DHA in these particular studies. So most of the DHA has to get made in the liver from alpha-linolenic acid precursors. So this is kind of the basis for what I want to go over today, which is looking in wild type and NRF2 at this idea of a gut-liver retina axis, and thinking about whether that could be helping to modulate this retinal degeneration phenotype. And so I'm going to show you data. We looked in 4 different kinds of parts of the mouse. I'm going to show you a little bit of gut microbiome data, where we did 16S sequencing and some pathway analysis. In the liver, we did RNA-seq, and we measured lipids. I'll show you a fair bit of work in the plasma looking at both some untargeted metabolomics and some lipidomics. And finally, in the eye, we did some transcriptomics as well, just in the neural retina.
Hey, Sheldon, could I ask a question? In the high glycemic and low glycemic diets, do they have the same composition of essential fatty acids?
Yes.
Good.
Yeah, I think the only source of lipids is soybean oil.
And it's the same in both.
It's the same in both, yeah. It's actually a fairly low-fat diet relative to certain other diet compositions.
Thanks.
So I'm going to show you, the high glycemic index diet ends up looking like a high-fat diet in a lot of respects, but the liver is doing all of the work in this case rather than coming from the diet. Okay, so I'll start with the gut microbiome. And one of the surprising things that we found when we looked in the gut microbiome, so as I mentioned to you, this is a whole body NRF2 knockout. And so we've also knocked out NRF2 in the gut, in the intestinal cells, everywhere else. We actually saw a huge impact of the genotype on the gut microbiome. Such that the genotype effect was actually larger than what we saw by diet, which is still highly significant. I mean, you can see that some of these dietary changes look like they're moving in similar directions. But then there's this also like interesting contribution of the genotype to the gut microbiome, such that you could see this is kind of like these are taxonomic bar plots, just looking at the family level. You could see like some of these families are really like highly prevalent in a wild type mouse fed a low glycemic index diet. And then you kind of go through this progression where there's less of it with an NRF2 knockout mouse, and then even less with a high glycemic index diet. And then if you combine them together, you've almost obliterated the amount of this microbiota. Whereas some of these guys in orange and green are actually dramatically increased in compensation.
I have a quick question.
Yeah.
Is So this is an assessment of distribution of these species. Is there also an assessment of the amount of each species? I mean, you're looking at fecal
Yeah,
pellets,
these
I
are
assume?
relative abundances.
Yeah, is there a good way of saying, like, for example, if there just are more microbes in the gut, a small bar might actually be equal, to a bigger bar if abundance is factored in. Um, is there a way of assessing that, or is that kind of, you know, or if you've done that, it doesn't matter, or what's going on there?
Yeah, we have looked at that, and there's techniques that you do in microbiome to try to deal with this, especially like with diversity. There's a process you could do called rarefication, where you like kind of look over. It depends on how many reads you get for each of the taxa. You can kind of like control based on differences in read number. We don't usually find a big difference in the actual amount of total microbiota.
Okay.
But you're right. You do have to keep it, these are relative abundances that we look at with the microbiome.
I've kind of a follow-up. The mechanism by which I think there are probably a lot of mechanisms by which gut microbes can affect, an organism. You know much more about the mechanisms than I do. But maybe even for me, certainly, but perhaps for the audience, can you state the main mechanisms by which you think that these gut microbe distributions are affecting the host organism?
Yeah, so my model is that I do think that they're gut microbial metabolites that are doing a lot of the dirty work here. And one of the classes, I'm not going to spend too much time talking here, but I think it's worth going back to. The gut microbiome has a huge impact on bile acids and circulation. So not just production of primary bile acids, but there's a number of secondary bile acids that depend on the gut microbiome to undergo these transformations. So the easy way to think about that is that the gut microbiome is the source of biosalt hydrolases that will take a conjugated primary bile acid, remove that, and then it can undergo further transformation. And these guys have really profound effects. And you'll see why I think they're important because they have a big effects on lipid metabolism.
No way. I'd love to hear more. Please go ahead.
Yeah.
Actually, I also have a follow-up question, sorry. Maybe you said it already, but are these microbiota derived from pellets, from luminal bacteria, or from mucus-associated
I'm
bacteria?
sorry.
And if so,
Yeah,
from which segment of the gut?
Yeah, this is fecal microbiota.
Okay, so ileal, for example, would have been probably more proper to detect changes in the bile acids, relevant bacteria, right?
Yes. I mean, we see plenty of BSH expressing microbiota in the fecal population as well. But you're right. If we measured the microbiome from the small intestine, we would see something looking quite different than this.
Okay, thank you.
Yeah, we have done some fecal microbiome analysis. It doesn't look hugely different. The fecal kind of being between the large and the small intestine. Okay. We don't need to go into this. This is kind of like another way of visualizing what's going on in the gut microbiome, where you could see we can do differential abundance analysis and find microbial taxa that are fairly genotype dependent. So some of these are only present in NRF2 knockout mice. Some we only see present in the wild type mice. We can see very strong dietary effects. So these microbiota go up with high glycemic index diets. These go up with the low glycemic index diets. And then we could see these interesting gene-diet interactions, where, for example, this microbiota is only present in the wild type fed a high glycemic index diet. And this is one of my favorite microbiota. This is a Bacteroides. You could see is almost completely depleted in those NRF2 fed a high glycemic index diet. And we can go and then take these kinds of compositional changes and then try to look at what are different pathways that those bacteria make and how could those microbial pathways impact host metabolism. What you can see here, I don't expect you to decipher too much of this, is that there's large networks of connected microbiota and their pathways. But you can actually see that actually a lot of the things that drive those pathway differences are related to smaller numbers of taxa. To the point being here is that when we look at the impact of the genotype and the diet on the microbiome, we actually see a much bigger impact in NRF2 knockout mice between the high glycemic, sorry, this is the high glycemic index and the low glycemic index in NRF2 knockout mice than we do in wild type mice. And so there really is a profound impact of both of them that needs to be considered when assessing host metabolism. I'm going to step away from the microbiome from now and move into the liver. And so what I want to show you are some of our liver transcriptomic results. This is looking at genotypes, so comparing the NRF2 knockout mice to the wild type mice. And you could see that actually the NRF2 genotype has a big impact on the liver transcriptome. This is not hugely surprising, but it is nice that we can kind of use this to identify what we think of as NRF2 target genes. One of the things that maybe surprised us a little bit, we saw 4 different main clusters here, but you can see that there's obviously like a big set here that's downregulated in the NRF2 knockout mice compared to the wild type mice. And these are where we see most of our canonical NRF2 target genes. So this is from a curated list of known NRF2 target genes. And you see that these mostly fall here. But we also have this huge cluster of genes that are upregulated in NRF2 knockout mice. And I don't think I appreciated quite how many genes would be upregulated in NRF2 knockout mice. And this is doing some gene ontology based on the clusters. And you could see all variety of pathways, more than I could possibly get into. But the areas where we see kind of the canonical NRF2 target genes in this cluster 4, these are the exact pathways that you would expect, things like glutathione metabolism and drug metabolism, for example. When we actually look at some of those major pathways, so these are kind of like our top 5 pathways that I picked here that, of interest. So drug detoxification, glutathione, inflammation, alcoholic liver disease, and lipid atherosclerosis. Again, what was surprising for us is that there's as many genes that are downregulated in the NRF2 as that are upregulated in the NRF2. And here's some examples of these target genes, and you could see the impact of genotype on those. The other way we can look at the liver transcriptome is by glycemic responses. So this is now comparing a high glycemic index in red here versus low glycemic index upregulated in blue. And we can now look at how the NRF2 genotype affects response to diet in the liver. What we see is, again, this idea that there's like very distinct clusters, such that there isn't a uniform glycemic index response. Actually, the presence of NRF2 has a really big impact on the dietary responses in the liver. And that was something that was surprising to us. I think you could visualize it really well here in this next slide. We kind of took the mitochondrial electron transport chain complexes where we found a lot of these genes. And what you could see is in the wild type, many of these are upregulated in response to a high glycemic index diet. Whereas in the NRF2 knockout mouse, not only do we not see these upregulated, but some of these that are previously upregulated in the wild type, here's an example of ATP5J2, actually becomes downregulated in the NRF2 knockout mouse. So that was an interesting finding to us, this idea that like these gene-diet interactions are really prevalent and probably quite important in the liver for a response to diet. And this is maybe starting to get at why we see some different phenotypes in the NRF2 knockout mouse compared to wild type in response to diet. And some of these genes, these are kind of broken up into a few classes of transcription regulators, cellular maintenance detoxification, lipid cholesterol transport. And you could see why I think it's really interesting to look at what's going on with lipids in the NRF2 knockout mouse in response to these different diets. So here's our kind of collection of fatty acid metabolism genes. I think these were curated based on the pathway. And what we can see is that some of these are genes that are upregulated in response to a high glycemic index diet in both the wild type and in the NRF2 knockout mouse. So a good example here is ELOFL2, which you can see is induced upon a high glycemic index diet kind of in a fairly genotype independent fashion. But there's other genes here that are only upregulated in response to diet in one genotype situation or downregulated in response to one genotype here. So again, these gene-diet interactions are also impacting lipid metabolism. So what's the net impact of all of these changes in lipid genes in the liver? This is where I think we're starting to see something interesting happen with the biology. So these are measures of the different fatty acids in the liver. What you can see here in the wild type, I've kind of told you this result already. So the MUFAs are dramatically increased. You can see we both see an increase in especially the 18-to-1 MUFAs in response to a high glycemic index diet. This looks fairly genotype independent. And this happens at the expense of PUFAs. So N6 PUFAs are decreased in high glycemic index fed mice. The N3 PUFAs are more decreased in the high glycemic index fed mice. So that was kind of what I told you before that was making me think that could be microbiota connected based on the transplantation result with the young versus old microbiota.
Quick minor question. MUFAs here are medium chain unsaturated fatty acids.
Sorry, monounsaturated.
Monounsaturated. Okay, great. And just making sure. Oh, and this is total lipids, like all together that have been fatty tails have been hydrolyzed off, or only free fatty acids?
I think it's total lipids.
Thanks.
Yeah. And again, like your question before, these are it's a compositional analysis. So these are not the raw amounts. These are normalized to the total lipids that we measured. That's why the DHA kind of falls off on the scale a little bit. But what we saw that was really interesting to us is not only do we see this drop in DHA in the high glycemic index diet, but the drop looks more profound in the NRF2 knockout mouse than in the wild type mouse. And to the point where normally you could see that in the wild type situation, we don't see a dramatic effect in the N3 to N6 ratio. There's decrease in both the N6s and the N3s. But this drop, especially in DHA and to a lesser extent in EPA over here, is such that we actually do now see a significant decrease in the N3 to N6 ratio in the NRF2 knockout mouse fed a high glycemic index diet. And that's kind of what I wanted to go with here. Like maybe some of our, retinal phenotype is being mediated by these alterations in production of N3 PUFAs in the liver. So that's what I have from the liver. The next thing I wanted to look at is what's happening in the plasma. So I'm trying to think about, again, this idea of like so gut, liver, retina axis. What's between the liver and the retina, or between the gut and the retina, is circulation. And like I told you, the gut microbiome has a large impact on host metabolism. And we really see that reflected in the plasma metabolome. So this is just an untargeted metabolomic analysis. But again, this to me looks almost like the same result we saw in the gut microbiome. So the difference in genotype in the metabolome is larger than the difference by diet. And again, we see the same kinds of trends. We can see metabolites. So now instead of genes or microbiota, we're talking about metabolites. So some of them are increased in the NRF2 knockout. These are GABA's actually increased. This is an interesting neurotransmitter that's modulated by gut microbiome. Is increased in wild type compared to knockout. We can see ones that are modulated by diet in both genotypes. And then we see, like, again, these profound gene-diet interactions where the levels of this phosphatidylcholine are really dramatically lower in the NRF2 high glycemic index, whereas this metabolite is increased, specifically in the NRF2 knockout high glycemic index. So you can kind of get a sense for how this combination of the gut microbiome and alterations in the liver are going to be impacting circulation. And here's, like I mentioned to you, there's a big impact of the gut microbiome on this metabolomic pattern. And what I wanted to do is I wanted to pick out, these are 4 different plasma metabolites that are dependent on the gut microbiome to make. And you can see that they're also impacted both by diet and by the genotype. So here's indole-acetic acid is a tryptophan-derived metabolite that depends on gut microbiota. Don't worry about all of the pathways unless you're really interested in one of them. I just wanted to point out 4 very different classes of gut microbiota metabolites that are all being impacted by NRF2 deficiency and a higher low glycemic index diet. And Daniel, you had asked me which ones I thought were particularly important. And I was like, I really am interested in the secondary bile acids. So here in the bottom right corner is one of these secondary bile acids. It's the 7-keto-deoxycholic acid. So first you need to make deoxycholic acid, which depends on gut microbiota mediated deconjugation of probably like a taurine conjugated cholate. So the TCA gets deconjugated, then you can make deoxycholate, and then it undergoes further transformation by the gut microbiome. You can see that this secondary bile acid is higher in low glycemic than high glycemic, but it's especially low in the NRF2 knockout mouse. So there's probably a combination of changes in the microbiome that impair production of this secondary bile acid. And a lot of these secondary bile acids are known to be neuroprotective. There was a gorgeous talk at ARVO by Maria Grant talking about one of these tryptophan-derived metabolites that seem to like have a very strong protective effect against diabetes in a model of in a diabetic retinopathy like model. But I also wanted to say what's going on with the lipids in the plasma. Given what I've shown you about impaired production of lipids in the liver, what's going on with diet? So this is kind of a more targeted analysis doing lipidomics. This is what we've published in wild type mouse. And this kind of goes back to one of the things that Jason was asking about is, do these mice become obese? They do. They also become hypercholesterolemic with elevated levels of LDL and HDL. And what I wanted to show you here when we do the lipidomics is that we see like a big change in the overall classes of lipids. So the high glycemic index diet seems to increase within the plasma levels of different phosphatidylcholines and cholesterol esters. Whereas in the low glycemic index diet, we see more of the lipids contained within diglycerides and triglycerides. The actual net levels of triglycerides are not different between them, but the individual composition are. So we did the lipidomics in wild type and NRF2 knockout mice. And obviously you could see that there's a profound impact of the diet on the overall lipidome. And we saw the same kinds of trends. So the low glycemic index diet, we saw these increases in diet triglycerides. Then the high glycemic index diet, we saw these increases in larger glycerophospholipids. The problem is, and this is where I kind of like, my knowledge becomes a limitation, is how to interpret all of the lipidomics. I'm not great at taking all the different species and assembling pathways from it. So I tried to do this, bear with me, using lipid maps, which you can look at different lipid species and how they're connected to each other. So this is basically like taking a pure network analysis of all of the lipidomics. And then what I did is I tried to look at which relationships are being altered by genotype. So ones that are in red here are impacted by, specifically the NRF2 knockout mouse. The ones in gray here are impacted in wild type over the NRF2 knockout. And then we could look at which pathways or which connections are affected in a high glycemic index state compared to a low glycemic index state. And I'll just quickly take you through a couple of the things we found from doing this network analysis of the lipids. One of them is that we found that, so here's looking in the high glycemic index state, the NRF2 knockout mouse seem to preferentially like to make C16 and C24 to 1 ceramides, whereas both the NRF2 and the wild type in response to high glycemic index diet make these 22 ceramides. And the low glycemic index diet, we see an alteration. Instead of going towards ceramide, there's a tendency to take those ceramides and make the sphingomyelins. So here's kind of what that looks like from a more box plot level. I mentioned to you the C16 ceramides are increased, specifically in the NRF2 high glycemic index, not so much in the wild type. We saw that same trend for the C24 to 1 lipids, whereas these are both increased in the wild type and the NRF2 knockouts. In the 24 to 0 ceramides, we didn't see a very significant effect of diet or genotype on those. So that's one class of lipids that's being specifically altered. Then I also want to kind of play this game of like, where is the DHA? And this is something that I struggle with a little bit, is like figuring out which are the DHA containing lipids. And if we're making less of it in the liver, do we have less of those different kinds of plasmid species in the plasma, and do those impact how they affect the retina? And what we see is different kinds of N3 containing lipids, some of which are these are the lysophosphatidylcholines are like basically a single chain compared to, this is just comparing phosphatidylcholines, which have two different chains, one of which may be an N3 chain compared to the single chains, the lysophosphatidylcholines. And this was a surprising result to me that hopefully one of you can tell me why. We see more of these in the high glycemic index condition in all of these different DHA or EPA containing phosphatidylcholines. So again, I would have predicted that if the liver is making less DHA, we should have lower amounts of these phosphatidylcholines in the plasma. But that's not what we see. But what we do see is, like, for example, I mean, I think this is a major DHA containing phospholipid in the plasma. There's a lot more of it in the wild type compared to the low glycemic than in the NRF2 knockout. So we do see some genotype differences. But maybe some of that could be explained by the different amounts of the diglycerides and triglycerides that contain omega-3 in them. So again, the ones that I've highlighted here are in orange that I could map contain EPA or DHA. And what we see is the opposite trend. So now the diglycerides containing omega-3s are increased in the low glycemic index compared to the high glycemic index. And so I don't know what the net ramification is on this kind of shift from the DHA going in the form of triglycerides versus other phospholipids in the plasma.
So I'm not strong in lipids, but I couldn't help but notice when you first showed the comparison of different classes that it wasn't just the LPCs, but also the PCs that were sort of universally up. When you had that plot of like a relative abundant, yeah, I have to go back even more, I think. Yeah, this one,
Yeah,
in D, I guess I was noticing that your LPCs were all above the gray line, and
right,
PCs
right.
were mostly above the gray line, and so I immediately had the question of, is there just like more choline there that would help to drive synthesis of that lipid class, of those two lipid classes, and just the very presence of that lipid as a sort of more abundant species could abstract, fatty acids that might normally reside on other species. I mean, I guess that's the question, even though it was stated as a, I don't know, a statement.
Yeah, no, that's interesting. So like, is it just precursor availability? I don't think I saw a change in choline by diet or by genotype. But I
Plasma
think it's,
choline or liver choline or both, or
oh, I never looked in the liver, right? So yeah, so that would be, yeah, that's something that's worth checking. I think I'm a little limited in only having done this in the plasma.
hmm. Similarly, your PEs are way down. Do you have LPEs in this? Oh, you do. They're not
Yeah.
down. Okay.
And actually, if we look at the classes here, there is a change here where I do seem to be forming more LPEs in the wild type than in the NRF2 knockout.
Yeah, too many groups and species and pathways to simultaneously wrap wrap your head around, right?
I
It's just so
know.
much.
I know, and the other challenge that I'm finding with the lipidomics is, I don't have anything great to normalize these two, and obviously the overall levels of these lipids like are much higher in the high glycemic index. So, this increase that I see here, in terms of the net levels, I don't know like what that really looks like. So everything's kind of like just a relative comparison.
Can't you use an internal standard with known concentrations for these different species and use that to quantitate?
I
levels,
have
not just
I have
relative.
some internal standards.
So
Yeah.
typically, I mean, I think some of the lipid experts on this call that I thought came in would know more, but I mean, typically you have an internal standard, at least one of each species, of each lipid species. And then if you're willing to make the assumption that under your mass spec conditions, you know, all fatty acid tails are coming off to the same extent and sort of, you know, 16O versus 18O, that they're all kind of ionized in the same way, then you can quantitate using using one internal standard for that whole class. Quantitate amounts, that is.
Yeah. I don't think I have tons of internal standards on this platform,
Okay.
but I
I
can
highly
see
recommend Avanti Polar Lipids. They sell these, you know, like mixes of internal standards for a bunch of different lipid classes as you continue to go about this.
yeah. Yeah, I definitely have to do a further analysis of the plasma to kind of answer that question. Okay, I'll show you one other result from the plasma, which is that we do see this interesting genotype effect on lipid mediators. So, you know, one of the questions is, are we seeing more evidence of lipid peroxidation in the NRF2 knockout? And I think the answer to that is yes. So some of these are clearly like oxidized. This is an oxidized EPA product. This is not an oxidized product. This is this is an icosanoid that's formed by lipoxygenase-mediated cleavage of arachidonic acid. And this is highly pro-inflammatory. And you could see that it's up in the NRF2 knockout mouse compared to wild type. But the other thing we saw are some other different classes of lipid mediators, and they don't know these mediators well enough to ascribe much biological function to them. But these guys, I think, are both considered to be adipokines. And you can see that they're actually up a lot more in the NRF2 low glycemic index compared to the high glycemic index. So there's so many of these, and we don't have a targeted panel to measure all of the different SPMs that can be produced. But I think that could be part of what is going on in explaining the biology of this diet effect. The last thing I'll show you really quickly, because I think it's almost time, is we did do the retinal transcriptomics. And actually, the take home from this was a huge disappointment. We didn't see a whole lot of changes in the retina transcriptome. I mean, we do see some NRF2 target genes that are downregulated. We see a lot more that are upregulated. And some of these came up in the liver. Some of them don't correspond at all. So I don't think like that what's going on in the retina is going to be explained by cell intrinsic differences driven by NRF2 genotype. I think it's going to really be mediated through these other impacts on lipid species and other, neuroprotective paths formed by the microbiome that are explaining that. So I'm done. I'm not going to summarize because it's short. These are all of the questions that I still feel are unanswered, that if any of you have insight into, let me quickly acknowledge everyone that helped with this work. Because this was an amazing collaboration with Daniel Brock and Anupam Mondal and Anand Swarup's group. Daniel is now an MD PhD student at Baylor, and Anupam's about to start his own lab, and a collaboration with Shifan Wang and Alan Ramali at the NHLBI. So it's been completely amazing. I wish we could understand all of the biology better, but I'll leave it here. If any of you have any quick questions before people need to get off the meeting.
Yeah, the floor is open. I know I've already asked my share, so I'll just wait for other people. Jim.
Yeah, I'm still wondering about the essential fatty acids, because if I remember correctly, you're saying that in the high glycemic diet, there were in the liver anyway, that I was remembering was that there were fewer N3 and N6 fatty acids.
That's right.
Yeah, and could it just, maybe this is just too simple-minded, but so when the amylose and amylopectin go into the gut, they get digested, and if the amylopectin is branched, so you get a higher release of glucose all at once, right? And the amylose is a single chain, so you get a slower release of glucose. And then you said that the diet influences the microbiome, the types of microorganisms that are there. And so isn't it just possible that when the glucose is released all at once, you get it selects for the microorganisms that aren't good at synthesizing essential fatty acids? Because it sounds like you don't have a lot of essential fatty acids in the diet, the N3 precursors for all the other, N3, N6 fatty acids. So is it possible that you're just selecting with a high glycemic diet, you're selecting for microorganisms that aren't really good at making essential fatty acids? And with a low glycemic diet, you get microorganisms that are better at making essential fatty acids. And could that account for the differences that you see in the liver and the plasma and everywhere you measured it?
Yeah, that's interesting. So the amylopectin is completely digested before it ever makes it into the gut.
But you still have a big burst of glucose coming through the system.
Right. There is a big burst of glucose. When we look by path, so I don't know how much of the fatty acid precursors that we use are coming from the microbes. I mean, it's not zero, but it's not a significant amount either. So I don't
know. You were saying it's a pretty low fat diet, right? You're getting soybean oil.
Yeah, I think it's about 10% of the calories are coming from fat.
Yeah. Well, anyway, it's a possibility. I just want to mention it. Yeah.
Yeah, no, I mean, I do think we have to keep in mind what lipids the bugs are making. When we do the pathway analysis, we see that it seems like the high glycemic index microbiota are more enriched in some of those the lipids biosynthetic pathways than the low glycemic index. So they may be busier making their essential lipids for their function. I just don't know how many of those we're using as building blocks ourselves.
Yeah, but the precursors have to come from somewhere in that.
Yeah.
Yeah.
Laura.
Oh, hi. That was a really wonderful presentation, and thank you. Always learning. I have a question relating to the hepatocarcinoma in the NRF2 knockouts. So would these different diets promote earlier onset, for example, or greater incidence of the oncogenic sort of pathways that would lead to either the gut colon carcinoma or the hepatocarcinoma? And would some of those transcriptome changes be in concordance with those pathways?
Yeah, thank you. That's a really interesting question. What we see is that there's definitely like a fatty liver phenotype with high glycemic index diets, and we've seen wild type mice. So we would expect an accelerated hepatocellular carcinoma progression, kind of going through, into cirrhosis. We did see a change in the prevalence, though, of hepatocellular carcinoma. It was prevalent in both dietary groups.
Right. Okay. So sort of equal.
So I kind of feel like maybe the inflammatory pathways were more relevant and more upregulated, such that you didn't need quite as much of the lipids to drive that, because there are other pro-inflammatory pathways that were helping to drive the carcinogenesis.
Right. An interaction between those two factors. Yeah.
Yeah, but no, I definitely expected more liver tumors in the high glycemic fat.
Right. I would have expected more, but you're saying there is not any noticeable overt change there. So
No, I mean, we didn't have a huge sample size, but I think the prevalence was similar between them.
right. And I had another quick thought. So there was no major changes in the retinal transcriptomic data, except there were some actually.
Yeah,
But
there
could
were
you?
some.
Yes, and it's a bit of a naive question, really, but is there a way of focusing more on RPE choroid type changes? Like if you were able to isolate that material, I know it just sounds really impossible, but if you focused on the RPE choroid, would that give a completely different context? And
I mean, I think it would. And, we have the RPE choroid because, I dissected that
yeah,
separately. So
yeah, it's
kind of
not
like that
impossible.
eye cup tissue. I wasn't super confident about getting a good transcriptome from
Right. How to
what we
extract it
had.
clean? Yeah, the RNA cleanly, I suppose. Yeah, yeah.
Yeah, I felt more confident that we had enough material to get a good retinal transcriptome. I do think there'd be more going on in the RPE choroid.
Right. Yeah, something possible anyway to.
Yeah, but I have kept it because I was also thinking like maybe that would be something that I want to do lipidomics on.
Oh, right. Yeah, yeah, yeah.
And unfortunately, I just don't have enough material to do both. I would love to
That's
do
it.
all
How
of
to do
it.
both. Yeah, yeah.
Yeah,
Thank you.
thanks.
So I'm going to be intentionally provocative, and I'm going to try to get you angry. I'm not going to try to get you angry, but so I think you had two statements that you made. One that was kind of at the end of your talk, and then one in response to Jim's question. In response to Jim's question, you were saying that you don't think that many fats, probably a small proportion of fats coming from the gut, are actually kind of feeding into or sorry, from gut microbes are feeding systemic metabolism. And then the other is that you think fats are, probably the more crucial element for the eye.
Yeah.
And so therefore, should you be studying in the future microbes?
I think I have to study microbes no matter what, because even if they're not like I don't know if they're a key source of precursors to make our fats. But there's no question in my mind that, their products are impacting those steps of metabolism and the different compositions of lipids that you make.
Hmm.
And that was again, that's why I picked the paper looking at the old versus the young um, microbiomes, right? It's the same mice, they're the same age, they just have different bacteria. And there's this really profound impact on which lipids you make in the liver. So I think they're impacting every process and every tissue. It's like almost impossible. But I don't think they're making like enough building blocks to contribute to those differences. I think they're really impacting more, um, the transport of lipids or the enzymes or other processes in making them.
But on
And
the other hand, I think at one point you were saying that they could be influencing the kynurenine and tryptophan pathways
Oh yeah, for sure.
significantly. And so if they can influence that maybe, they could also be influencing the amount of essential fatty acids, because otherwise we have to get the essential fatty acids from something, right? And those guys, I'm sure they have all the enzymes to make those. And so maybe they're pouring out N3 and N6 fatty acids, like the way they would with the kynurenine and tryptophan.
They could. I mean, I guess I think the dietary fats are completely essential. Like, I don't think the microbes can make enough of them. So, like, if we didn't have like alpha linolenic acid in the diet, I don't think we could make any DHA or EPA. So, I get maybe I'm not understanding the question.
But somebody made the alpha linolenic acid in the first place, and so it's usually bacteria or algae or something, right? And so if our guts are full of these little microbes that can make it, why not just
Oh,
use them?
oh, I see. I don't think our commensals make it. Like, I
Oh,
just don't think we have the right bugs to make those. Maybe that's what I'm misunderstanding. Yeah. it would be interesting, right, if you could modify the microbiome to like produce more of them,
Right, right, right.
and then
Yeah,
see
I
how
just assumed that all bacteria could make them, but maybe I'm not. Maybe
no, I think it's
yeah.
a pretty specialized group of
Okay.
that makes them. And
Okay.
that's why it's so limited to like these aquatic
Okay.
organisms. Yeah. Oh yeah, thank you. Now I understand.
Yeah.
Hey, Sheldon, I have two questions. I, there are so many moving parts on this. If the
Yeah, no
NRF2
kidding.
knockout, the diet, whether it's from the gut, whether it's from the liver, what's going on locally in the retina. Uh, and I guess it's slightly provocative, but are there controls when people do gut microbiome studies to figure out whether it has what's actually happening in the retina, the phenotype in the retina has anything to do? You can see lots of effects in the gut and lots of effects, on the liver, but that may or may not have any impact on the actual phenotype you read out in the retina. For example,
Right.
I mean, I don't know, humans can live without their colon, for example. I don't know how, how do people control for that the effect is clearly coming from the gut microbiome rather than something else? Because you could put in old gut microbiome in a new, in a young mouse and vice versa, but does that stay and retain over an entire lifetime, or do you have to sort of gavage them all the time? I'm just trying to figure out the ways to figure out the effects you see, the phenotype in the eye is truly coming from, a difference in gut microbiome.
Yeah, no, those are really important and hard questions. So the best way to do that is work with germ-free mice, right? So if you work with germ-free mice, and Dimitris Gandra's group is doing this, but not in an aged context, then you can completely control, do they have any microbiota? Or you could make them have a very specific microbiota. Um, and there's no question that germ-free mice have impaired everything. Like the immune system is messed up in germ-free mice, liver metabolism is altered in germ-free mice. Like all of those things are different in germ-free mice. And then you could either reconstitute them. So what was nice about Marianez's experiment is that when you start with the germ-free mice and you put in the young versus the old, that's their microbiome, right? Like it'll change around based on, as they age and competing impacts of the diet. But I mean, you've basically like, given them their set of commensals. What I've tried to do in other experiments that I've not presented here is I tried to swap the microbiomes from from the high glycemic index and the low glycemic index. And like you say, doing those experiments are a mess because as you feed them the diet, the microbiome keeps changing. And so what I've done is I keep giving them, I've gavaged them week after week after week with the microbiome to try to change it. You end up with like an intermediate result. You end up with a new microbiome that contains some character of one, but something that's unique to it because it had the old microbiome in there as well. So the best we could do is we could just change the microbiome without changing any other factors. So give them the same diet throughout the study, but all we've done is change the microbiome through these fecal microbiota transplants. And when we do that, the eye outcomes definitely change.
Okay.
So it's the gut microbiome can direct those. Like what it does to direct them, I mean, could be like any pathway.
But the bottom line is the swap experiments have been done, and because I thought I saw with the swap stuff you were looking at plasma and liver, but I was wondering about what's happening in the eye with the swap.
Yeah. No, in the eye, it largely works. So like if I give my high glycemic index mice a low glycemic index microbiome without changing the diet, um, they they protect most of the eye. It's not perfect. The weird thing is they still become obese, um, but their glucose metabolism's improved. So, I'm not sure what's really contributing. Like you say, there's just so many moving parts that it's hard to tease out one factor alone. So,
Well, then, I mean, this seems to be the essential point of the research question is like, how do you deal with all these moving parts? How do you get them to fit into the same framework?
Yeah, no, but it's a good point. You just try to change as few parameters as possible. So, like for example, one of the experiments I would love to do, I've mentioned to you a bunch of times, I think secondary bile acids are important. But what if we give them bacteria that are engineered so they don't have those genes anymore, so they can't actually do the bile acid transformation? Um, do they still show neuroprotection, right? Like, so try to change one thing at a time, even though everything else is still going to change
Yeah, yeah.
with,
I mean, there's this interaction, especially when you're bringing Nerf 2 into it. There's this
yeah, yeah, yeah.
whole series of interactions, and it's so. But that's biology. I mean, I'm not asking
Yeah.
a new question. I'm just kind of, you're dealing with this, I think, more than I have to deal with this. So it's just interesting to get your perspective.
But I do think like it's a general problem for those of us interested in metabolism because there's so many pathways that
Yeah,
do get affected.
they all
It's really
interact.
hard to be targeted in any single treatment. But I'm interested, like for example, would DHA be sufficient to rescue some of this phenotype? Because if it is, then I could be like, okay, all those other things may be important, but the DHA is actually what's limiting. And by putting more of it in, we can rescue this without needing like a low glycemic index diet.
Yeah, I think that's great. That would be
It
great.
won't rule out though that adding DHA to mice also changes their microbiome. So it's like you still can't disentangle everything, but you can try to do like a little bit of reductionism here and there.
All
I
right.
have another question. So first of all, thank you for bridging finally the microbiome with ophthalmology. I come from the one field, switched to the others, and I
Oh,
think
that's
it's time for
awesome.
this. Yeah. So one connecting or several collecting non-intestinal diseases that, for example, are associated with IBD are usually uveitis, rheumatoid arthritis, and so
Right.
on. And so far, the main connecting factor, as far as I'm concerned, is systemic inflammation. I
Yeah.
remember Maria Ursinio, for example, has also shown that when you have penetrating bacteria that come from the gut, obviously, you also have a temporal opening of the blood-brain barrier. So have you ever checked for either inflammation in the gut or inflammation in the eye, which could explain the phenotype you're seeing?
Indirectly, I have tried to do both of those things. The one thing that I'm not good at measuring at is systemic inflammation. I don't have good results when I try to look at cytokine levels, for example, like in the plasma.
Or CRP or something like this, SAA?
I've tried CRP. I don't think I saw any consistent effects on CRP. I do think there's a lot going on locally. Um, and I'm trying to look actually right now. In this study, I don't think we saw like a strong inflammatory signature in the retina. I do think in our last study, like looking at the retina transcriptome, we did see like a bit of like a pro-inflammatory signal from the high glycemic index diet.
Mhm. Or maybe LPS in the blood or something like this to potentially
Yeah,
see any breakthrough bacteria.
right. Yeah, no, I mean that's kind of like the appealing model, right? Is breakdown of the barrier and then release of LPS or other products. I know no, we've tried to measure the LPS. We haven't had success. I think I could try to do LPS binding protein to look at it. But I don't think that's what's happening in the gut. I don't think it's like necessarily a breakdown of the gut barrier.
Okay.
But I do think that in the retina, it's different. I do think like the RPE barrier function is really altered.
Yeah. But
And
beautiful multiomics study, definitely.
thanks. It's really hard. I'm still trying to connect things because there's so much to try to connect.
All right. Does anybody have any final questions? And if not, I thank you so much, Sheldon. This was a wonderful talk, and I appreciate you taking the time to give it.
Oh, thank you so much. And everyone that stuck around, I appreciate it. And if you have other ideas and want to shoot me an email, I'm really like open to ideas for this. It's still like a fairly nascent project. So
Yeah, cool. Yeah. All
Hey,
right. Thank
Dan,
you.
do you, I was
Oh,
just going to quickly chat with Sheldon afterwards, but do you have to close this thing off?
uh,
I can just,
I don't know. Oh, yeah, I mean, you chat, stay on.
okay.
I'm gonna
you can
stop the
stop. Can you
recording.
stop